Today, scientists meet the future scientists. Are you guys ready for it? Let's go! Hi everyone! This is Eunji, and I'm currently a third year majoring in psychology at UC Berkeley. Today, I'll be going over a topic called How the Brain Works and provide further details about our brain, including myself, Professor Lee from a neurology department at Ewha School of Medicine, and Soo-hyung Kim, a medical student from Ewha University, are in our research team. With our research, we have created a very interesting video that can make our young friends grow as future scientists who will explore brain science as part of projects by the Organization for Human Brain Mapping. So these are the following key questions that we'll go over in this talk. First, how can we see the world? Second, how does the brain communicate? Third, how can a computer think like a brain? Last but not least, can we see how the brain is working? Keep these questions in mind and listen carefully what I'm going to say. And I'll ask you all these questions at the end of my talk. Do you guys like our world star BTS new song? What comes to your mind when you hear this BTS song? Can you imagine how the brain would react after listening to this song? The word BTS stands for our idol group called 방탄소년단, but it can also imply brain transfer stimulus. It's the most crucial function of our brain that transmits stimulus. Soon after this video, the word BTS would not only remind you of the idol group, but also the brain transfer stimulus. Let's get started then. Do you guys wonder how we can all see? It's all because of our brain transfer stimulus. The brain transfers any visual stimuli, so whenever we see or hear something, as you can see here, our brain conveys very important information about our vision and hearing, right? Interpretation of human vision. Neurons are key workers in our brain. Our brain consists of more than 100 billion neurons in the brain cortex. The primary visual cortex are orientation selective and interprets as the simplest form like dots, while V1 represents only the most basics of visual features. The visual cortex processes more and more complex information as you go farther in depth. In the secondary visual cortex, the neurons are responding to a little more of visual patterns than V1. As we go farther in the visual cortex, the neurons in the tertiary visual cortex and other associated visual cortex interpret more complex and very specific visual information, such as our friend's face or beautiful scenery, etc. Using this information, we know that there is a correlation between the levels of complexity of the different parts of the visual system process and the levels of complexity of what we're seeing. Organization of human visual cortex. In the brain figure on the left, the posterior part of the brain marked in red is the visual cortex that is responsible for vision. As soon as we see an object, the visual cortex immediately analyzes information. It only takes 70 milliseconds. Whoa! Can you imagine how fast that is? Millisecond means 1/1000 second to transmit the information to the primary visual cortex. At 100 milliseconds, the visual information is transmitted to the secondary visual cortex, and at around 120 milliseconds, it goes to the tertiary visual cortex. As a result, Brain signals of specific waveforms at different frequency bands are recorded at each level of visual cortex. Brain transfer stimulus connectome. Before, I talked about research that focuses on different wavelengths in the visual cortex that functions interactively. As I mentioned earlier, there are approximately over 100 billion neurons in the brain. These neurons are the nerve cells that process information by sending electrical and chemical signals. The neurons interact with each other in a very complex way to process information. The brain contains an overall brain mapping that represents a connection among nerve cells. Just like a subway map that connects all the subway stations, connectome demonstrates a link between neurons in a brain. In case you guys are wondering how the connectome looks like, this is the following picture of connectome. Isn't it so pretty? On the left side, we have the first picture of connectome. From the article called The Mind of Worm, the researchers drew a clear picture of a little nematode's connectome. And for over 10 years of research, they cut the worms in 50 nanometers to analyze the data. And here on the right side, from the National Institute of Health Human Connectome Project, by using various methods such as fMRI and PET to see a cross-section of a brain and completed the brain mapping. By visualizing the neural network, we can spot any abnormal connection parts in the connectome and diagnose a particular brain disease or analyze related factors. Many research are currently being conducted on how much the connectome is like in normal or abnormal brain conditions. Artificial intelligence is a computerized algorithm that mimics these neural networks in the brain. The picture on the left side only shows a small scope of a brain's neural network, and the picture on the right side is a computerized algorithm. Do they look similar to you? In fact, like a brain, these artificial neurons form a network by combining synapses, and through sending and receiving signals, they produce problem-solving skills. We perform computerized vision or speech recognition through supervised and unsupervised learning, pattern recognition, etc. With computerized vision, we can recognize any disease that is difficult to detect with our own eyes. And the speech recognition in this case implies speech-to-text systems such as Siri on Apple and Giga Genie on KT. Artificial intelligence is getting popular in brain research, and it is also useful in interpreting the brain's network. We can build an AI-based brain network model using this artificial intelligence. Using the electrodes onto the brain, we measure the brain signal, so-called electroencephalography. Applying it to AI brain network models can help us figure out whether this is diseased brain or not, whether it is normal or abnormal brain function through artificial intelligence. Before I introduce our interesting research, I would like to briefly go over the functions of the brain. Our brain has four main lobes: frontal, parietal, temporal, and occipital. The front part of the brain, frontal lobes, are mainly responsible for speech and motor function. Located on top of the ears, the temporal lobes are for understanding language and auditory function. The parietal lobes on the side of the brain are for sensory and calculation, and on the back of the brain, occipital lobes are mainly for vision. Our last question: how can we see the brain works? Is it true that the brain is divided into different sections for each function? How can we check that? We use various ways to actually observe how the brain works. In the brain, the interconnected neural signals send information through minor electrical signals. If these normal electrical signals are released as abnormal, seizures can occur. In order to check this, Just like the picture on the right side, we directly implant the electrodes into the brain and measure the brain's electrical activities. The picture on the far right side shows the brain with the surgically implanted electrodes on the surface. Other than that, as I mentioned earlier, we use brain imaging tools like fMRI and PET to interpret the connectome, and from the brain structural or functional brain imaging as we analyze the brain's network. The brain waves generally show regular and continuous fluctuations. If the person is internally excited, the amplitude of the brain waves is small and the frequencies become irregular. If you see the picture on the right side, in the case of a convulsive brain waves, you can see irregular frequencies. Even in the case of an abnormal brain, brain waves with unique characteristics are shown. So if we carefully analyze the brain waves frequencies, amplitude, and waveform, we can easily diagnose seizures or degenerative brain disease. Electroencephalography, or EEG, can be tested even without physical harm to patients. So it is widely used to precisely identify abnormal areas of the brain. Another research that I want to talk about focuses on how the brain reacts differently when we locate electrodes on the patient's visual cortex and give a series of visual stimulus. When the patient perceives this blinking red light repeatedly as visual stimulus, the electrodes recorded a different spectrum of waveforms. And from the spectrum analysis, we can interpret the spatial temporal components of visual functions. The picture on the right side indicates the different waveforms recorded from each electrode on the brain surface, marked in blue, as we interpret information from the visual stimulus. This study compares the frequency that appears promptly when we place the electrodes on the medial, inferior, and lateral cortical surface when the visual stimulus is delivered. I'll explain further details about the results from the medial, inferior, and lateral brain surface, that is, the specific cortical locations where the simple form or intermediate visual response or complex visual information like face are interpreted. Just like the picture on the left side, the blue dots are where the actual electrodes are located, and the fluctuation graphs below show the corresponding frequencies of the electrodes. Before, A has shown regions of the brain that react differently for each visual stimulus in the picture. Do you guys remember the primary, secondary, tertiary visual cortex? When we give a simple visual response, like the basic form or shapes of an object, an intermediate visual response with slightly complicated forms or patterns, or a very complex form of visual response with a complicated form, colors, and movements, we have different levels of visual information. When we have simple visual responses, the blue parts of the electrodes are activated, and when we have intermediate visual response, the cortical areas with green triangle parts are activated. For complex visual responses, the areas where the red stars are activated. The figure B below is the spectrum analysis of the visual evoked potentials that appear based on various visual stimuli. When we get a simple visual response, we have the fastest response, while in a complex response, the reaction appears slow and lasts for a longer duration. I'm going to show you guys one video here. Do you guys see any changes when you watch the video? As you can see here, the redder it becomes, the more this area becomes activated. At a high frequency, it reacts and fades out rapidly, like in the primary visual cortex. And at a slower frequency, you can see the brain activation appears later and lasts longer in higher visual cortex, like secondary or tertiary visual cortex. In this study, We directly place the electrodes into the brain and examine how the brain responds when we give different visual stimulations every time. Do you guys remember all the questions that I asked earlier in the beginning? Let's recall all the questions and the answers. First, how can we see the world? It was because of our brain transfer stimulus. The brain transmits the stimulation, and this helps us to see. Next, how does the brain communicate? In a brain, there is a brain mapping called connectum, so we can see how the neurons are connected to each other. Third, how can a computer think like a brain? It's called artificial intelligence, and it mimics a brain's neural network. Lastly, how can we see the brain is working? By recording and interpreting brain signals, we discover the ways the brain works. What word comes to your mind when you see the word BTS? Does it still remind you of 방탄소년단, or does it remind you of brain? T stands for transfer. And lastly, wait for it, wait for it. S stands for stimulus, brain transfer stimulus. If that's the case, yay, we have reached our goal. Isn't it so interesting to know how the brain works? There's still so many things that we haven't figured out about the brain yet, but we have our future brain scientist right here. So let's take part in brain research as a future scientist. Thank you guys for listening to my talk. Bye!